Okay, I see us on YouTube then, so that's done. I'll tweet it now. I think everyone's doing the classic thing where you turn up 30 seconds before it starts.
I mean, the only reason I'm on time to these things is because I'm, streaming it.
Yeah. But it's 4 o'clock, so I'll get started now and then, yeah. Okay. Hello, everyone. Am I on microphone? Yeah. Okay. Hi, everyone. Welcome to this week's Cell Migration Seminars. We're going to do a slightly different format for the next month, so we're going to have a lot more seminars where we have a couple of speakers rather than just one. If you have any feedback about that, like whether you prefer just one speaker or a couple, then get in touch and let us know. And then the other thing I have to say is just a reminder to ask questions. So on YouTube, you can just type your questions in the chat and either me or Jen will read it out. Or on Zoom, you can type your questions in the chat, or preferably, if you'd like to ask the question yourself, just write the word question in the chat. And then when we call your name, you can turn on your microphone and your camera and ask the question yourself. We very much encourage you to do that. So it's not just us asking the questions. But that's pretty much it. So I'll get on with introducing the speaker today. So our first speaker is Brian Camley. Brian is an assistant professor in the departments of physics and astronomy and biophysics at John Hopkins University. Brian's lab studies the physics of cell biology with theory and computation, with a particular interest in collective cell migration and chemotaxis. And today, Brian's going to be talking about contact inhibition of locomotion on suspended nanofibers. So,
all right, so let me go ahead and share a screen.
And the rest of us can stop our videos and mute ourselves.
All right, so are you seeing the correct screen now? Or are you seeing?
Yep, we see you. It's not in presenter mode, but we see your PowerPoint.
All right. So that now you're seeing.
Brian, just to let you know, we can see your slide, but also the next slide and any notes in that mode.
All right. I'm going to just unplug my second screen.
Okay.
So, sorry, I'm used to doing this without PowerPoint.
No problem.
Just a quick reminder for everybody to mute their microphones during the talk.
Okay. Are you seeing the screen not in presenter view now?
No, we don't see anything now.
All right. How about now?
That's perfect.
All right.
Thank you.
Thank you.
Great.
Okay. So I think I'll go ahead and try to get started here. So thank you for the invitation. So I'm happy to talk about some recent work with Amrinder Nain, and just in general, a little bit about contact inhibition of locomotion and some of the modeling that my group has been interested in doing that. So everyone has, I think many people have heard of classical contact inhibition of locomotion, or CIL, where cells that are traveling along collide with another cell and then repolarize away from that contact and then move away. So it's sort of in a sketch here, or in this nice nearly one-dimensional collision event from Roberto Maior's group. This is with neural crest cell explants. And this, it goes back to the 50s and the original discovery of this, and its contact inhibition of locomotion is an important element of normal development. So what you're seeing here, once I start playing this movie, is Drosophila hemocytes, and you can see that as I play, you'll see the nucleus moves out. The nucleus here is labeled, and you see that the hemocytes manage to spread out. So you can imagine, relatively easily that, something like modulating spreading or something like that without direction might be something that would be mediated by contact inhibition of locomotion, but it has significantly more important roles as well. For instance, when cells work together in order to sense a collective signal, you see here on the left a group of cells, again, neural crest cell explants following an SDF1 gradient signal, whereas single cells don't follow that. And it turns out that contact inhibition of locomotion is essential for this process and how it occurs in the Xenopus embryo as well. And the reason for this is that as cells are oriented away from contact, you see that orientation away from contact allows them to be more polarized up the direction of the chemoattractant, and then there can be an effective tug of war between cells. And it's a little bit more complex than this, and I've actually spent a good amount of time modeling this. So this is actually one of the things that I spent a lot of time thinking about, but it's not the subject of this talk. So please just take a look at this review, this review that I've cited over here, if you're interested in this. What I'm going to be talking about today are more about collider-like experiments. And, I call this a collider experiment sort of as a joke. My background is in physics. And so it's always wonderful to say, oh, okay, how do we study cell-cell interactions? Well, let's collide a bunch of them together and see what happens. And so this sort of interaction is actually it's a statistical property. You can get a range of different outcomes. So at the top here, we see a repulsion, a sort of classical contact inhibition of locomotion for these neural crest explants. That's what you see about the majority of the time. Sometimes, however, cells will just stick together and not manage to separate and repolarize, even if they do repolarize. And a very rare fraction of the time, about 5% of the time, you see walk past, where one cell manages to completely walk past the other and separate. And even if you inhibit contact inhibition of locomotion, this walk past event is usually pretty rare. Other cell types can have other behavior. What I've shown you so far is neural crest cells, but if you look at epithelial cells again in this quasi one-dimensional geometry where they're on a stripe, you see that there's sort of an asymmetry between front and back, where cells will end up, if they collide with one another, they'll end up forming a long train and all polarizing in the same direction. So you don't see that same characteristic separation behavior in all cell types. So what I'd like to ask in general is, what physical and biochemical features control the outcomes of cell-cell collisions, and what can modeling tell us about what's essential in these cases? So one of the elements that we know from earlier experiments is that contact inhibition of locomotion is linked with RAC repolarization. So here on the left, you see a movie of a single cell showing RAC activity, indicating the cell front. So we see that in this case, it's following a gradient. You see a lot of this RAC activity at the front. However, at a point of cell-cell contact, you see something where originally you have high levels of RAC activity. After then the cell contact, RAC activity drops, and then RAC, which would normally indicate the front of the cell, repolarizes to the back. Actin polymerization follows that, and then the cell ends up crawling in the opposite direction. So the sort of qualitative picture that we would have in mind from a model like this, from an experiment like this, and earlier experiments from Roberto Maior's group and others, is something like contact repolarization, where two cells contact each other. There is a local inhibitor of RAC that's activated in both contacting cells. And then as a result, the RAC repolarizes and the cells separate. So this is something that we can put together as a computational model. So the basic elements of this model, this was done with my postdoctoral advisor, Wouter-Jan Rappelle and Dirk Kulalwiak. We put together a nice model of where we include the RAC activity levels over the entire cell. That's this red color here, indicates where RAC activity is high. That indicates the direction of the cell. And then we have protrusion where RAC is high and contraction everywhere else, which sets the shape of the cell. So we track the boundary of the cell and the evolving shape. And we also include then something that inhibits RAC within the model. This would be upregulated at contact, but we also allow it to just sort of fluctuate to allow the cell to have a fluctuating path so that it's a persistent random walk. And if we run this simulation, what you see is that you see, you simulate the cells, they come together, they contact, and you see RAC inhibitor generated in both cases, and then RAC repolarizes away from the contact, and then cells separate. So this is something where we can, with some relevant physical and biochemical forces, recreate this contact inhibition of locomotion. However, one of the things that we had seen was that in different cell types, there's some evidence that there is some asymmetry between the front of cells and the back of cells. So if you made a slightly different hypothesis and you said, well, suppose then that RAC inhibitor is generated only in cells that touch the cell front. So in this interaction between the head of one cell and the tail of the other, you only get inhibitor here in this sort of front repolarization type model. Then if that's the case, you'd expect both cells to continue and to form a train. And what you get is then, again, if you simulate that same initial condition where where cells run into each other head on, there's a spontaneous symmetry breaking where only one cell ends up repolarizing and they form a train. This is something where we can then explore other factors that make a large role in controlling the outcome. And in fact, we can reproduce all of the known two-dimensional contact inhibition of locomotion types just by playing with, so we can switch between reversing and chains by increasing front repulsion versus contact repulsion, and we can transition between reversal and sticking by increasing adhesion, and we can also actually create walk past. However, this worked very well for these quasi one-dimensional substrates, but motility in extracellular matrix and in sort of biologically relevant contexts isn't like motility on substrates. This is something that has been accepted and sort of understood and studied in a large different degree of context, and we'll hear another talk relevant to this later today. But you see here on the left this keratinocyte crawling on a 2D substrate, and you see this nice broad lamellipodia, whereas if you're in matrix, you see a more elongated shape, really persistent motion, and you see something similar on sort of 1D microstripes. And there are biological consequences as well for roles of things like myosin and microtubules that are different between 2D and 3D. So a question then is, how does this fibrous extracellular matrix geometry alter contact inhibition of locomotion? And this is work where, I'm a computationalist. All of the experiments have been done by Jagrup Singh, who's in Amrinder Nain's group at Virginia Tech. And this has just been posted on the archive, so you can find that So the idea here is to use a controllable extracellular matrix that models some of the features of a true extracellular matrix. So we have this sort of fibrous structure, but instead of having, you know, something like a collagen matrix, we Amrinder and Jagrup create these aligned fibers and coat them with proteins, and they're actually suspended above a substrate. So they're lifted up off of the substrate. So cells aren't in contact with a substrate at all. They're only in contact with a fiber. So what happens? If we look at contact inhibition of locomotion on these single fibers, 2/3 of the time, cells walk past one another, almost completely ignoring one another. So this is very different from the classical stereotype of contact inhibition of locomotion. This happens in 2/3 of the time, whereas in the neural crust experiments, we saw this at maybe 5% of the time at most. The remainder of the time, we see behavior where cells impact one another and then start going in the same direction. So they form little trains. That happens about 30% of the time. So if you look at what happens on these fibers, what we see is that only 4% of the time do we see the sort of classical CIL reversal type behavior. So this indicates that there's something really different going on. Division is also a really important signal in these experiments. So if you look at cells that have recently divided, their behavior is a little bit different. So watch the division, and then we'll see a walk past happening again. So if you quantify this, you see that when cells that have recently divided, walk past happens 82% of the time, up from about 2/3 without division. And the other important aspect of this is that after division, cells speed up quite a bit. So you see that there's a huge difference in the mean speed, although again, there's a lot of variability. Now, one of the things that we saw, you know, in this extracellular matrix, we were looking at sort of these fiber models are you know, only approximating a real extracellular matrix. Obviously, you could have cells attached to more than one fiber. You have a lot of interesting little dynamics of what's going on. So what happens if we bring our fibers a little bit closer together? So instead of having this elongated spindle shape that you see on top here, you see that they can attach to two fibers in a parallel shape. So what happens for cells in this parallel geometry? Well, then they impact and then they form this nice train. And in fact, that happens all of the collisions that we saw. It's only 28 collisions, but we see just a huge amount of training in these parallel collisions. Again, this may reflect the fact that there are fewer degrees of freedom for cells like this. But what happens if we throw division into it? So if we throw division in, then cells, even in this parallel geometry, are able to walk past one another. And in fact, it's a pretty big effect. It's the majority of cells walk past one another, up from 0% without division. So what I'll try to give you then is just a brief summary of the experimental picture. There's a lot more in the preprint, but the brief summary of what I think are really key results are that, unlike earlier work on stripes, even normal cells, not things with TGF-beta or contact inhibition of locomotion inhibited, walk past at high rates in the spindle geometry. The parallel geometry, where you have two fibers, suppresses walk past really robustly. And when you have division, it speeds up cells and it increases the rate of walk past in both the parallel and in the spindle geometries. So what we'd like is to know, to what extent are just the physical changes in the geometry that you're attached to two fibers instead of one sufficient to explain the altered contact inhibition of locomotion behavior? So that's something that we can try to address with the physical model. Now, in this case, because it's a three-dimensional problem instead of a two-dimensional problem, I tried to reduce the complexity of the model to one that's a lot simpler. In fact, you know, again, I almost have to say, as a physicist, this is your stereotype of me, but I'm going to start representing these cells as spheres. And what we're going to do is write a description for the cell polarity. So the polarity here is going to be a single vector that represents all of the details of the dynamics of all of the rack and the inhibitor that we described in the earlier model. So this, and the idea here is that the cell velocity will be equal to the cell polarity. So polarity would be the same as velocity if there weren't any other forces on it. That indicates the direction and the speed the cell would like to take, but then there are forces on the cell because it's attached to the fiber and it's in contact with other cells. So let's describe what the forces on the cell have to be. So one important thing in understanding this is that you can recognize that looking at the microscopy here, you can see that the cell body can be tilted to one side of the fiber or to the other. And so it's important to include that in our model. So when we described the cell fiber forces, and I'm going to describe this in some amount of detail so you can understand the assumptions here. So we think of the cell being attached to the fiber by basically just a little spring force. But the important detail about this is that instead of having something where the equilibrium position of the spring is at the fiber, it's that the equilibrium position of the spring is at the cell radius. So it likes to sit to one side and can freely rotate around the fiber. Then the cell-cell forces, we have one element of cell-cell forces in the model because effectively for cells to come, the center of masses of these cells to come close to one another, the cells have to deform. So cell stiffness opposes overlap between cells. So it's like having a repulsive spring there. And cells that are within an adherence distance become adherent to one another. So that's keeping them together. So there's a force keeping them together. But once they cross that distance, that adhesion ruptures. So again, a relatively simple model. I told you what the forces were. Now let's focus on what the cell polarity is. Again, this is trying to summarize all of that rack dynamics into one simpler model. And so what we're going to do is we're going to write an equation of motion for the cell polarity. And what's going to happen is we would expect that the cell polarity would relax with some time scale tau, which is going to be a persistence time, how long the cell keeps its characteristic velocity. And it wants to relax its polarity to where it would be comfortable, basically. And the idea of comfort here is that, if this were a cell that didn't have any signals on it, is that it would relax to just a random direction, but instead there's this contact guidance. Cells tend to polarize along the fiber. So we create this capital P, which is either plus the x hat direction, that's along the fiber, or in the opposite. So you're either traveling along or opposite to the fiber. And if you start out pointing in this direction, you tend to go in that direction. So then on top of that, we add a little bit of stochastic noise. And then on top of that, we add contact inhibition of locomotion. Contact inhibition of locomotion, of course, has to include all of these features that we talked about before, so it's going to need another slide. If we assumed that cells just repolarize from contact and don't care about where the front of the other cell is, there's no asymmetry in the contact behavior, then the natural choice would be that if we look here at this second cell on the right, that it would be polarized away from the direction of contact. However, if we care about where the front of the other cell is, we use the front repolarization mechanism, which is what we're going to use. That's the natural mechanism here. Because the cell polarity is pointing away from this cell, we say that there is no contact inhibition of locomotion on this cell. But if this cell to the left reorients, then this cell is going to be repolarized and tend to be repolarized away from this cell. So that, again, is just trying to summarize all of those complex rack dynamics. So we can run a quick simulation. And what you see is that the cells do manage to walk past each other if they're attached on a fiber. And one of the things that you should see about this is that the cells aren't, sort of phasing through or pushing through each other. What you're seeing is that they are on different sides of the fiber. So effectively, it's not obvious in the first movie I showed you experimentally, but cells can walk around each other. and you can see this in the experimental movies where you see that a cell rotates in order to move around the cell that it's impacting. If we then put 2 fibers in, you've removed that degree of freedom, and cells instead manage to train up. So you see that that's reproduced nicely and fairly robustly. So let's see what we can do in terms of comparing to experiment. So the experiments showed that walk past happens commonly in the single fiber geometry. In the 2 fiber geometry, it suppresses walk past, so it sounds reasonably good. And division increases both speed and walk past. Now, one of the difficulties in comparing to experiment is that even in a simple model like this, we have a lot of parameters. We can set 6 of them, things like this typical speed and the geometry from the experimental data, and we vary the rest over a plausible range. So what we do is we take a set of parameters, which we choose randomly from the plausible range, and then we run about 200 collisions with it, just head-to-head collisions, and we ask what fraction of them walk past. And what I'm circling here with this red circle is the value for just one parameter set. For one parameter set, you see that the walk past fraction is about 75%. But if you choose another parameter set, you would get another dot in this dot graph. So what you see is depending on the parameters, you get different rates of walk past. So if you increase the adhesion, you get less walk past, things like that. But no matter where you choose the parameters from a plausible range, you really commonly get walk past in this spindle geometry. So far, so good. If we ask about the effect of the parallel geometry and we take the spindle walk past fraction and we subtract off the walk past fraction in the parallel geometry, again, each of these points is for one parameter set. We see that for every parameter set that we choose, when we move from spindle to spindle to parallel, the difference is positive. So the walk past rate decreases from spindle to parallel for all of those parameter sets, so really robustly. So that's good. But then we have one leftover question, which is division increases both speed and walk past. And I haven't told you how we could reasonably model division. And so we can make a hypothesis, which is that the effect of division is just increasing the initial degree of cell polarization, or the initial speed of the cell, basically. So if that's the case, instead of starting out a cell that's polarized with one characteristic polarity, we started out with about 3 times that polarity. That's something we can estimate from experiment. So if we do that, we see that we can get walk past behavior even in these parallel geometries, and that if we compute the change in walk past fraction for all parameters, again, so in the spindle geometry, we take the rate in walk past for the spindle cells with division minus the spindle cells without division, and we can see we always get more walk past in the spindle case. And in the parallel case, we usually get more walk past. Sometimes we get 0 change in walk past. That's because if you have something that's so adherent to both fibers that it can't possibly deviate from its position, there's no way to have walk past. So the walk past rate effectively always goes to 0 in that parallel geometry. So that says increasing polarization increases walk past. So what I'd like you to take from this is that cell-cell collisions and CIL on nanofibers can be qualitatively different than on 2D substrates or even on microstripes because of the additional degrees of freedom that are available to cells on fibers, and that can be modulated by changing the geometry of fiber structure. We can capture these differences in a really simple physical model. And the physical constraints and things like the cell speed from cell division can completely change outcomes. All right. So thank you for your time. Again, as I said, the RAC and the RoGTPase models with deformable cells was done with my postdoc advisor, Wadrian Rappel, and by Dirk Kulawiak. The CIL on nanofibers work was with Jagrup and Amrinder. This paper with a lot more details about things like cell speed and so on is right now just on bioRxiv. So thank you.
Thanks very much, Brian. If you'd like to unmute and give a round of applause, please do. So I'll ask some questions along with Jen. So I'll start with one from Santosh Priyal asking about how you think the RAC inhibitor partitioning is achieved in the cells, and whether RAC inhibition at collision sites could generate an active pool of RAC, how that happens somewhere else in the cell.
I'm sorry, could generate an active what?
So will RAC inhibition at the collision site generate an active pool somewhere else in the cell? So how's that partitioning achieved?
Okay, so at least within the level of the model, what's happening here is that this RAC, I'm using a model for RAC polarity called a wave pinning model that's done by Leah Edelstein-Keshet and a couple of other folks. One of the things that's interesting about this model is that it will lead to spontaneous polarization of a cell. So effectively, if you inhibit the polarity at contact, the cell will choose another direction. The particular direction that it chooses is going to be dependent on sort of what else is going on in the cell, including the geometry of the cell. So in our case, it tends to just move directly in opposition. All of the different factors that are going into that, I think, are not as well known.
We've got a question here from Anna Huttenlocker, who asks, with cell division, is this because the contact inhibition of locomotion after division between the dividing cells is the dominant influence / dominant polarization and not the new contact event?
Yeah, so that would certainly be consistent with what we're seeing. Again, I think we would have to be a little bit careful about it because contact inhibition of locomotion, sort of this, again, the stereotype is that like you're polarizing away from contact, but we don't see that with cell-cell contacts. We only see that with dividing cells. So I think it's saying that the polarization event from the cell-cell division is a much stronger effect than than the cell-cell contact later on.
I have a question from Leander Burr asking if you've imaged the RAC dynamics and whether the increase in speed in the dividing cells could be due to an unequal distribution of the RAC between the cells.
No, we haven't, or I can't say that I've imaged anything. Amrinder and Jagrup have not done any of that yet. We would really like to. I think it would be a great thing to do. I think some of that has been done in colliding cells by Andrei Levchenko and Takanori Inoue and Ben Lin, which is one of the papers I cited earlier, but we haven't done it, not in this context.
Great. We'd like to invite Roberto Mayor to ask a question.
Hi, Roberto.
I have a very simple question. So the contact inhibition that you analyze in these fibers is you analyze it assuming that this is happening in one dimension, but this is really a three-dimensional thing. So if you have two cells that are colliding, as you explain and is in the model, the cell can go to the other side of the fiber, and that is why maybe you have. So my question is, have you analyzed? So maybe still in these fibers you still have contact inhibition, but the contact inhibition will be that when two cells are in the same side of the fiber, the cell will move to the other side in order to pass work. So how you analyze if this kind of event is really happening? And this maybe is the way in contact inhibition is happening in these fibers.
Yeah,
not repolarized in a way.
I agree. I think that it's sensible to think of this as sort of like a different way in which CIL can be realized, as opposed to specifically saying that it's absent. And that's certainly in the model. The question of effectively with the images that we have, it's not always obvious where the cell is on the fiber. So we've had trouble tracking that. That's something that we would need to do either some degree of confocal stacks or something like that in order to, I feel, get a conclusive answer about that. And that's something that we haven't done yet. But I think we do see these nice experiments where it just happens to be in the correct plane that we're imaging, where that's what's going on. So I'm pretty confident that that is what is happening.
Okay, thank you.
Question from Tony Tsai asking about why you think the cell moves faster after the division.
So again, I think actually one of the earlier questions got to what I think about that, is that effectively the cells are really immediately polarized by the division event. Many different signals in division are going to be on one side of the cell versus another. You have all of this sort of new membrane that doesn't have the same set of signals on it. You have microtubules distributed to a different location. We don't know whether it's driven by contact sort of interaction between the two dividing cells or not. I will say that this is something that another group, Bojana Gligorovic, I believe, saw something related to this in one-dimensional confinement, where you saw it, depending on cell phase, different speeds. So that she also may have something to say about that.
Okay, great. We've got a question from Parag Katira, who says, as always, cool modeling. And then two questions, actually. So we've just got about a minute left, so we'll do these quick. First, should fiber diameter matter, and how would it get incorporated into the model? And then the second question is on cell division. It seemed from the experimental videos that the actual cell, the cell actually leaves one of the two fibers. And could that be the reason for the increased walk past? The increased polarity might actually be dragging the cell along one fiber more than the other in the model and recreating this.
Okay, so unfortunately I've forgotten the first question already, but the
fiber
second
diameter.
Oh, okay. Oh, fiber diameter. Okay, yeah. So it does matter. You'll see in the preprint that depending on the fiber diameter, you see very different lengths of cells. So that will affect the contact, though we don't have systematic data. Some of them were a lot easier to image than others. I don't know how it would. It wouldn't show up physically in the model. It would more be that it would modify the relevant parameters. But it's definitely an interesting area. In terms of the fact that you see that the cell leaves one of the fibers, that's absolutely true. We don't have a model for that. We expect that to help to increase the rate of walk past beyond what we have put into our model. And so something where we study the shape in a more dynamic way, I think would capture that beyond sort of this very simple model.
Thanks very much, Brian. That was really great. There was a couple of questions on Zoom and one on YouTube that we didn't have time to get to, but if you'd like to address them in the chat or on Slack, then
yeah,
do.
so I'll try to address them on chat, on Slack, or get to me on Twitter or whatever. Thank you very much.
Great, thanks a lot.
Okay.
So moving on to our second speaker today. Our next speaker is Anne Reversat, who is a postdoc with Tobias Zech's group at the University of Liverpool. Anne did a PhD in the group of Anna Maria Lennon at the Curie Institute in Paris, before moving to a postdoc with Michael Sick's lab in IST Austria. And today she'll be presenting the work she did in Michael's lab. The title is Cellular Locomotion Using Environmental Topography.
Yes, so thank you very much to give me the opportunity to present my work. So indeed, as you said, I'm in Tobias Zech group here at the University of Liverpool, and I will show you the work I did. So as a postdoc in Michael Sick's group at IST Austria. And this work is about, sorry, I want to reduce this, okay, yeah. Is about the role of the environmental topography in cellular locomotion. Yes, so first of all, this work was really recently published. So we'd like to thank all the co-authors for the great work that we did. And that they did also. And so what we are interested in. So we are interested in leukocytes that are really fast migrating cells that need to move towards the whole body. So they are generated in the bone marrow, and they develop, then they migrate toward the lymphoid organs, such as lymph nodes, and they need to patrol towards the whole organism within tissue in the search for pathogen to protect your body against invasions. So cell motility is essential during your whole life for efficient immune system. And so how does the cell migrate is the question we want to address. And so this is the classic view of cell migration. So as you know, so cell move by a mode that is called aptokinetic, which means that they need to hold on the substrate when you put them on 2D surfaces. So what you have is that the cell here that is polarized. At the front, you have actin, filamentous actins that polymerize. And then you have transmembrane proteins, such as the integrin, that link with the extracellular matrix through a cytosolic adapter. So here, the tallin protein. And this allows the couple of the actin forces with the substrate. And this makes the cell move forward. So what happens on 2D when you have no adhesions? So you can have no adhesions either by removing tallin, the molecular clutch, or removing the adhesion molecule, or even by changing the extracellular matrix to one that your cells are not adhesive to. And what happens is that then actin still polymerize, and you have a really high retrograde actin flow from the front to the back of the cell as it cannot couple with a 2D surface. And what about leukocytes? So leukocytes are extremely fast migrating immune cells, so they move by a mode that is called amoeboid. This means shape changing, and they have really low adhesive force. So when you look at them, they're weak, short-lived integrin contacts, even in 2D, and they move by this then shape change. So actin polymerization at the front and contraction that is dependent of the myosin at the back. And you can see one of the things about those cells is that they need to migrate throughout the whole body, into different kind of tissues. So basically, in vivo, they move mostly embedded into 3D structures, so either like in small lymphatics, for example, or embedded within other cells, or even embedded into some tubular matrices. And most interestingly, they are way faster in 3D than on 2D surfaces. And more than 10 years ago, so Michael Sick's group showed, so this is the work of Tim Lemmerman and the collaborators, and they showed that if you remove all integrins, so all adhesion protein from leukocytes, so this leukocyte are dendritic cells, you can see this is the integrin knockout cell, and this is the wild type cell, and they are indistinguishable. So basically, leukocyte can migrate without any adhesion in 3D environment. So they show this both in vitro, in 3D collagen gen, but also in vivo. So what are these mechanisms of adhesion-independent locomotion in 3D? So what we decide to do is that we decide to remove the molecular clutch by removing the adapter protein tallin. This is because leukocytes express numerous integrins, but they only express one gene of the tallin protein. So we did this using the CRISPR system. So you can see here that we managed to get some cells that do not express tallin. So this is an antibody against both isoform. So now we generated cells that cannot translocate the forces from the actin polymerization to the extracellular matrix. And how do they look on 2D? Well, it's quite simple. If you look at them, the control cell, they just fall on your plate or on your dish, and they can stick a bit to the bottom plate. And the tallin knockout cannot at all. They are really round and they are really floating. So this was really first striking observation that shows us that this indeed inhibition of the molecular clutch work. And we further confirm this by looking with a specific integrin ligand, so FcICAM1, how the cell can spread on 2D. So you can see here you have the control cell, here the tallin knockout cell, and here you can see the epifluorescence, and here the TIRF that show you how much the control cell here can spread onto specific adhesive surfaces, whether tallin deficient cells cannot spread, they just round up and are floating there So we then check how they migrate in 3D, and what is good is that it's the same that Michael group shown more than 10 years ago, is that tallinn knockout cell, tallinn deficient cell, are indistinguishable from their wild type, from their control adhesive counterparts. So you can see here cells are embedded within a 3D collagen gel that you cannot see the fiber, but they are embedded in 3 dimensions and they can move really efficiently. So that's started the actual question: how can those cells that have no adhesion migrate in 3D? And the first hypothesis that they could use the confinement of the environment. So when they are embedded, they are squeezed by the environment. And this was shown already from the 90s that so neutrophil, if you squeeze them between 2 parallel surfaces, they can migrate really fast by a mode that rely weakly on adhesion called shimney. And this was further investigated also in other type of cell. And so to do this, to look at the role of the confinement, I developed a confiner from Mathieu Piel lab in Michael group. And we also developed with Jack Marin a quantity of microfluidic devices. And why did we do this? Is that it's true that in vivo the environment is really complex, and it's really interesting to look at cell motility in this context, in the natural context. However, it's really difficult to control the microenvironment. So using this confinement or microfluidic system, you can really control the geometry, but also the coating, the chemical coating of the environment. So how does the cell look when they are squeezed here between 2 parallel plates separated by 5 micrometers? So here you can see the control cell. So in red is filamentous actin, in blue is the nucleus. And I think you can really well assess here that the control cell migrate really efficiently within the confinement, whereas the tallinn depleted cells are really inefficient. So they can really not use the confinement to move forward. They're really running on the spot. And they are sleeping on the substrate. And when you look more carefully at the actin cytoskeleton, how it looks, so here you have control cells. This is the actin in filamentous actin, standby life like GFP, and this is the tallinn knockout cell. You can really see that the cell run on the spot. You have filamentous actin polymerization at the front that is flowing backwards. And here you have again the control cells that can migrate here from the, and the tallinn knockout cell is really slippery. So this is a kymograph where you can see here the cells that migrate, the control cell, and here you can observe that actin can translocate with the substrate, and it's not the case with the tallinn knockout cells that are running off the spot and present a really high actin retrograde flow. So this was really important result because this shows that even if people have shown before that in confinement, cell can move really fast using low adhesive forces. If you have no adhesive forces, like in our tallinn depleted system, the cell cannot move. So how can they move? What we decided to do is that we added some topography within this confinement. So we added first micropillars here that you can see that create a kind of forest, a pillar forest that is easily controllable. And how does the tallinn knockout cell look like in this topography-bearing environment? Well, they look like this. So this is a tallinn knockout leukocyte. So you can see here filamentous actin in black. So you can see that the cell are really look like a wild-type cell. You have actin protrusion at the front, and you have the cell can really move here. So you can see this is a bright field image. You can see how well it can move between the pillars. We further quantify this, and what we could see is that control cell can migrate in all different zone where we have different spacing between the pillars here, but you can see that Tallinn knockout cells, adhesion deficient cells are really become less efficient when the distance between pillars increases. And this this shows that basically they need the topography of the pillar forest to move. So we decide to explore this further by increasing the surface contact that is between the cell and the environment. So what we did is that we use now microchannel. So we first use smooth microchannel where we can find the cell, and here you have a control cell that can migrate into the microchannel, and you can see how the Tallinn knockout cell is really inefficient in migrating, so it cannot translocate any force. We further quantify this. And the most importantly is that if we had some small local topography, like if it, like for the pillar forest case, so like if it would imitate some topography from the 3D environment, like for example collagen bundles. What we can see is that here, so you can see this is a Tallinn knockout control cell. You have here a channel with a different topographies, and here you have a zone with a smooth zone, and the control cell can migrate in all cases, whereas the adhesion deficient cell can only migrate in a microchannel where there are a topography, where there are topographies. Okay, so we also used another system to show this. So we use chemical depletion. So that's a control. The motility is restored in serrated channel. So we further use a chemical system to deplete adhesion. So we use EDTA that chelate bivalent ions. So this removes adhesions. And you can see that in simple confinement here, the cell, they stall and they slow down. They really have difficulty to move. Whereas in zone where you have topographies, the cell migrate quite efficiently. And this was further quantified here. So you can see that on in confinement, or in smooth channel. So this is here in gray. The cell have great difficulty to move with no adhesion, whereas when they are in the serrated region, cell can more efficiently use the topography to move. So the big question is, how can a cell migrate via topography? So to look at this, we collaborate with Raphael Botturier and Andrew Cala-Jones, who are physicists, and they made a physical model. So we simply modeled actin as a viscous gel that travels from the front to the back of the cell. And this viscous gel then flow backward. And this flow has a length L. And what happened is that here they introduced in a model topographies with this distance here. So the distance between two topography. And what happened is that when the channel is smooth, the flow moves from the front to the back with no problem, and there is no generation of friction because there is no adhesion. And when the topography are introduced, you have the generation here, the flow is basically bent by the topography, and so you have a generation of perpendicular, of shear stress of the actin that is bent, and you have perpendicular forces that are generated. And this induces a pressure gradient of this force within the actin gel cortex, and this allows the cell to move forward when those topography are smaller. The distance between two topography is smaller than the length of the actin flow. So this was a nice. We wanted to see if we observe actually this, whether the cell migrate because the topography are smaller than the length of the actin flow. And so what we did is that we designed channels that are topographies with different spacing, so 6, 12, and 24 micrometer. And I think that you can nicely see here that the control cell can migrate throughout the whole channel, whereas the tiling knockout cell here migrate efficiently in the topography when the topography are close by, but when the topography are spaced, so around the length of actin retrograde flow, the cell have difficulty to move forward. So this was further quantified in a region with a lot of close by topography, the cell can move forward. And in highly spaced or topographies or smooth channel, the cell cannot migrate. We further looked at, so Julian Stock looked at other cells, so neutrophil cell, and Ingrid De Vries look at dendritic cell. Yes, so this is neutrophils, and you can see that this is a control cell. They nicely migrate within all zones of the channel. But when you add EDTA, you can see here that, like we saw with our other T cell model, the cell cannot migrate within the big space substrate. And now you see here dendritic cell. On the left is dendritic cell with adhesion. On the right, dendritic cell with no adhesion. And you can see that there are really difficulties here to move, whereas they can move efficiently here in the zone with where the topography are nicely spaced with a smaller distance. So we further looked at how the actin behave within these different zones. And here you can see a cell, a T cell line migrating under non-adhesive conditions, so where we add DTA. And you can see here that you have a really impressive flow from the front to the back of the cell. The cell have really stalled and difficulty migrating forward. However, this cell managed to turn and go past it. And now you can see that when it's at the zone where the topography are close by to each other, we see no actin retrograde flow. There is a direct clutch of mechanical clutch of the actin flow with the topography, and the cell can move. This is another example, so the cell can move nicely in these two zones here. And then when the topography are highly spaced, you can see here that the cell cannot efficiently move and has this really high retrograde flow. So here I forgot to say this is the time in minutes and seconds, so leukocytes are extremely fast, so you can see that we can move within all this, within the first zone in the cell speed that is around 10 micrometer per minute, and then in a 24 or smooth zone, they really cannot migrate and they present this high retrograde flow. Yeah, we also looked at TIRF, and we see the same behavior of the actin cortex at the TIRF level. And finally, I would like to show you the work that was done by Florent Gartner, and this was also further analyzed with the help of Saren Taskian. So what Flo did is that he embedded this time primary T cell under agarose pad, where he coated the bottom surface with specific integrin ligand, so IKM1. Otherwise, he coated this smooth surface with PEG, but he also uses REACH that he coated with PEG. So PEG is polyethylene glycol. It's a chemical coating that removes adhesion. You can see here the orientation of the REACH. And what you see nicely is that primary T cell can migrate easily under confinement with a specific ICAM1 adhesion here. And when you add the non-adhesive chemical coating, what happens is that the cell cannot move. So in the same case that for chemical depletion or genetic modification of adhesion, so the cells are really running on the spot and cannot migrate efficiently. And most impressively, in this ridge bearing confinement, what happened is that the motility of the cell is restored and is restored perpendicularly to those ridge. So meaning that the force necessary to move forward generated by the actin cytoskeleton is perpendicular to the ridge as it's implied in the physical model. So they further looked together with Robert Hochschild, they did analysis how this retrograde flow looks. This is a cell under smooth peg coating, and this is a cell under the non-adhesive, so peg coating, but the cell is put on top of ridge. And you can see here, I think it's really nice, you can see the flow of actin that is coming along the ridge, and you don't see flow coming perpendicularly to the ridge, so they further quantify this. So they measure using optical flow and spot tracking how actin behave. And what they can see is that indeed, along here, the topography, the actin flow and cannot couple, whereas perpendicular to the topography, there is no flow because the cell can couple the polymerization force with the substrate and migrate in this direction. Okay, so I think I was extremely short, so that's good, and then we'll have a lot of questions. To summarize, this we show for the first time that leukocyte can move in 3D without any adhesion by coupling directly the cell actin flow with the environmental topography. So this explains why cell without any adhesion when they are on 2D or in confinement cannot move, and then when they are added into artificial topography bearing surfaces or in 3D structure, whereas in collagen gel or in vivo, then they can migrate forward. And this is really important for leukocytes because leukocytes, as they migrate into various tissue in vivo, it makes them completely autonomous in their locomotive behavior from the composition of the environment. So in the sense that they don't need the specific adhesion molecule to go throughout the whole tissues and then react really, really fast. So I would like to thank everyone. So this is people from the 6th group. So all this was done at IST Austria with this funding, ERC grant, and FWF fund, Austrian Fund for Life Science. So I want to thank Michael, of course, Ingrid, Yolan, Miroslav. Yeah, so Mirek did the primary T cell. I think I forgot him. So Saren, Flo, Jack Marin, and Juan Aguilera from the nanofabrication life science facility, Robert Hochschild and all people from the imaging facility. And also, so this work was done in collaboration with physicists from Paris. So Raphaël Voiturier from the Laboratoire Physique Théorie de la Matière Condensée and Laboratoire Jean Perrin from the Université Pierre-Marie Curie, Andrew Callow-Jones from Université Paris-Diderot, Laboratoire Matière Système Complex, and Mathieu Piel from Institut Curie and Pierre-Gilles Vogène. So if you want to reach me now, I'm in Liverpool in Tobias Egg Lab. So here is my current mail, and you can also reach me by Twitter. Thank you.
Thanks a lot, Anne. Please unmute and applause. Thank you. Great, lots of questions. So we'll get started. First one from Ankita Jha asking, do the talon knockout cells form more lamellipodia-like protrusions under non-adhesive and confined environment? And is contractility lower in the talon knockout cells?
So sorry, the first beginning was, do they form more protrusion?
Yeah, in the non-adhesive and the confined environments.
I didn't notice this, at least. For me, especially, they look still polarized with one protrusion. Then if they have higher contractility, I couldn't really see it. For example, when they are confined, they bleb at the same efficiency than the wild type cell. Yeah, so yeah, I don't think so. What we did is that we did, we did the experiment with blebistatin, but mostly with the EDTA treated cell. And so here we could see that this migration is not dependent on myosin, in case this is the question. So we still have a migration that is dependent of the zone with topography. That is only happening in the zone with the topography when we have the blebistatin. Even if the migration in general is reduced, so migration speed is reduced when we have blebistatin, it's still happening.
Great talk. So Pragati on YouTube is asking, how do you check the adhesivity of the cells?
So that was the first slide. So basically we checked them by coating specifically on 2D surfaces and then measuring how much they can spread, and literally sotalinocote cells cannot spread. They are just round blobs that fall, and then we measure the TF area there. I also did it by counting manually. Yeah, that's how we measured it.
Joshua Grolman asked, does the curvature of the confinement make a difference?
Yeah, so that was so we have some data for this. So we tried, we didn't. So I'm not sure exactly of the question. So it could be that if the curvature indeed, I think it's the case in the model, if the curvature tend to be smaller, so not as sharp, the cell will migrate less efficiently. However, we didn't really measure it quantitatively because it was quite hard. And yes, so I don't know if this answers the question. After we also used channels that have sharper side, for example, more triangular, and the cell can still migrate efficiently when After there is also a limitation in the production, so it's always a bit wrong. So.
Thanks. Leanne de Boer asks, do the cells change in volume as they're confined in the channels?
So no, I don't know if it's in the channel versus versus confinement, and I don't know if it's if the question is also do they change depending on the adhesion. So this I measure the apparent like surface area, and they do not change. Perhaps you notice that there are under confinement, extreme confinement, especially some cell looks bigger with big confinement because then they have more problem to divide. So then we didn't use a big cell to quantify cell motility, but otherwise, no, we didn't see change in volume. So I think it's around 300 meter cube. I cannot really remember. I measured it, so yeah.
We have a question from Alessandra Camby.
Oh, yes, thank you. I was just going to leave because I had to leave, but I have time now. Very nice talk. Thank you very much. I was just curious about, do you think there is any role for the glycocalyx in the type of friction that you see there in this locomotion, especially in your topography environment?
So I'm not sure about the friction, the role of the glycocalyx in the topography. So like, why wouldn't this work, for example, just in smooth channel?
Yeah.
So that's a bit tricky. What I want to tell you is that in a classic confinement, so in this pushing of the wall system, I strongly believe that there is a role of the glycocalyx because when the cell the cell are really sensitive to changes in the serum or system. I don't know how this would exactly work. So yeah, I would imagine. I don't know if the glycocalyx. I would imagine that it will also flow, but I don't know. Or, yeah, it's a good question.
Okay, thank you. Thank you.
Akankshi asks, is there any relationship between the length scales of the topography and the cells?
So this we looked, so it's the relationship is really between the length scale of the topography and the length scale of the actin flow. So if the cell has a big actin flow, we would imagine that it can migrate with using the topography that is all embedded within this actin flow. And we actually measure the cell length, and the cell length is way higher than the actin flow indeed. And so no, we don't believe it's a full cell length. And after when I measured, I measured manually, and the bigger cell don't have the bigger outflow length. So I didn't see a correlation between these. So you can have a really big cell that have really really lame actin flow from the front to the middle of the cell. Yeah.
Anna Huttenlocker asks about the width of the channel. So does the channel width influence the movement of talon knockout cells, or is it just the topography? And might it be a case of a defect in squeezing into small paces playing a role, like less contractility?
Yeah, no, so we don't believe that this is the case because, for example, I don't know if it's after, but we did, oops, sorry, no, I don't know. We use, for example, a stronger confinement, and there the cells are really highly blebbing in both cases. So it will not be a defect in contractility. I don't remember the first part of the question. I think it was also whether what happened if we increase the width of the channel, but this is basically what we are doing here. This in confinement, this is like an infinite width channel, and also flow. So the work that Florian Gartner did on the topography surface also shows that you have basically an infinite width. The cells are confined and they can migrate perpendicularly to this small topography. So, yeah.
We have a question from Angelica Gasolina, who asks, how do you decide on your topography distances or ridge shapes? And then can you comment if these mimic any specific tissue or organ?
Yeah, so actually our widths here, the width we use are the one that is found classically in tissue. So in general, it's not really constructive. So here we are above, I don't know if also this is a question, but we are above 3 by 3, for example, constriction, where there you have really a high constraint on the nucleus. So we are in the range. So I think it's 5 by 5, 25, 30 micrometer square. I think it was work from Peter Friedel and also Jörg Renkewitz. Yeah, so we are within this average range of constriction. So we are not really in extreme range. And sorry, the second part, can you what I cannot remember.
Oh, just if there's any mimicking of any specific tissue or organ.
Yeah, no, we don't really mimic any tissue after, for example, yeah, no, we don't really mimic any tissue. I think we mostly mimic what we saw in collagen gel. Yeah.
We've hit 10 past. I just want to check with Anne. Are you okay to stay for another few minutes just to answer some more questions? Yeah. Okay, great. So I'm just going to combine the next three all together. So a question about how is actin flow coupled to nucleus deformation in confinement, and then a second part, different thing about how unique this is to leukocytes versus other cell types like fibroblasts, or can this mechanism might apply to other cell types?
Yeah. So we didn't look at all at other cell type. I mean, in our model, it's something that could work with any type of cell. Yeah, so we don't know yet if this can also work with fibroblasts. I think perhaps some people did some work where they put DTA on collagen gel, and there's perhaps the fibroblasts couldn't really make it. I'm not so sure about it. I cannot really recall. So yeah, but however, we believe that this is kind of a universal mode of migration. Yeah, as long as you have this higher retrograde flow. And then concerning the nucleus, so we have really no role of the nucleus in the model, but it's true that, for example, the nucleus could have a role in constraining the actin cortex next to the substrate, for example. So perhaps this will allow like to have a cortex that is really close by to the topography. So I didn't see, so sometimes this was done mostly with a T lymphocyte. I know that dendritic cell in certain conditions, they can cut and have a cytoplasmic parts with no nucleus. So it will be interesting that to look at this indeed, to see if like you could have a flowing membrane that just migrate with no nucleus. Yeah, I don't know.
Amrinder Nain asks if there are geometric conditions under which cells reverse direction.
So here, both in the model and in the cell type I used, I have actually highly polarized cell. Even when they're in suspension and you look at them, they have one front, the lamellipodium at the front, they don't have multiple protrusion, and they have the back that is contractile, so you can really see it already when they are floating. When they are, sorry, so it's about basically polarity and changing. So we have actually, we did some experiment where we have, what's the name, ratchet-like channels, so channels that are orientated. But we saw in both cases, funnel kind of cases, and the other way basically, we saw that cell can migrate with the topography in both directions. So after, whether there will be one mode that will be more efficient than the other, we didn't really test it. In the model, it's not the case. It's different from this polarity mode. So most of the time when the cell was going one way, it was never going back. So I mean, a lot of time it was intrinsic cell polarity that made them change.
A question from Wan Zhang-Lin. Since the flow in the microchannels is subject to Poiseuille's law, I'm sorry if I'm getting that name wrong, is there a chemical gradient inside the channels?
I don't know if you ask if there is a chemical gradient of for example, chemokine. For all this that I showed with T cell was done with no chemokine. However, we could also see this result with a primary T cell with chemokine, but there was no gradient within the channel. So it's true that this could be a question, like if there is a gradient on the other side, would the cell be forced perhaps to repolarize? This I don't know, but no, we didn't use gradients. So after, I don't know if it was also gradient of other molecules that he was thinking of. Like we don't have also gradient of adhesive protein or, yeah.
Okay, we have a combined question from Claudio and Robert Insall. So Claudio Franco wants to know if you think the cells require topography all around the cell, or the topology just in the leading edge, whether that's sufficient for motility. And then Robert wants to follow up on that and ask, in the movies, it looks like cells stop once their front third is not in the ideal place. And is that a true observation?
This is a really true observation. So what I didn't show you is that most of the work done in the Tallinn knockout cells is done with a microfluidic device that uses valves to push the cell in the structure, because otherwise they cannot migrate. They need integrin to migrate within complex environments. So if you put them, yeah. So I think Michael is still working, Michael's group, Ingrid has some data on it. So the cells are not efficient to invade either 3D structure or smooth or parallel structure. You need adhesion for this indeed. So it's not flow only coming from the lamellipodium. It's really, I don't know after if it's because the flow length is too short. I wouldn't, yeah, I don't think so, because then they will be more efficient in collagen, put on top of collagen. But here at least they need flow that goes after the protrusion, that goes, yeah, up to the back, the contractile zone of the cell, from the front really to middle of the cell.
I think this is the last question that's related to what you were just talking about. Akanshi's asking about whether the cells prefer cavities of certain types of topography.
Sorry, excuse me, can you repeat?
Whether the cells prefer cavities of certain types of kinds of topography.
Yeah, yeah, I'm not sure. Like, perhaps the question is whether they will prefer to go into zones where the fibers are more or less oriented. This we didn't test, so it will be quite, I mean, question that we could generate this actually. Some people could, I guess. Yeah, so for this, yeah, I don't know. They just need to have topographies that are passing through the cell, whether it's collagen bundles. Yeah, I guess that it will work better when they are perpendicular versus aligned, because that's what we saw on topography bearing surface. But otherwise, yeah, I don't know whether which type of environment they would prefer. Yeah.
All right, so this is actually the last question. Robin Saul just wanted to follow up on his previous and ask, it looks like the pseudopod needs to be on the correct topology, not the cell. Is that correct?
I'm not sure about what does he mean about the pseudopod?
Rob, do you want to jump in and ask if you're on?
Yes, just one of your really beautiful videos in the middle showed the sort of rapidly changing actin-rich third of the cell exploring the topography. And then when it changed, when you went from the rapid change to the slow change, once the pseudopod was in the slow change and the cell body was in the rapid change, it didn't move anymore.
You mean it stalled a bit at the entrance when it changes the two different zones?
That's right. It seemed like the secret with the pseudopod was not in the topography. Is that right?
Yes, it's true that this is something we saw. Yes, so I don't know if it's due because indeed this topography what's the name number like change. So then the flow doesn't couple as efficiently as before. But it's true that we saw that the cell stalled a bit and then managed to move forward. Yeah, it's true. Yeah.
Thank you.
Yeah, you're welcome.
Great. I think we've covered all that. So thank you very much, Anne. That was a really great talk.
Yeah.
And thank you everyone for joining us. If you have any further questions, I think Anne and Brian are both on the Slack channel, and I think they're both on Twitter. So feel free to follow up on either of those platforms. And we'll see you all next week.
Thank you.